So the peptides that we're dealing with here are peptides that are capable to generate amyloids. Amyloid molecules in general are what happens when proteins misfold. So they're usually a result of something problematic that happens in the cell. So when a protein misfolds, many times it creates long fibers that deposit in the cell and cause a lot of diseases that are called amyloid-related diseases. So there are actually quite a lot of very well-known diseases that are associated with amyloid formation, such as Alzheimer's, type 2 diabetes, and other very well-known diseases, which share the occurrence of amyloids, but usually not a lot else in common. Okay, so in the recent years, there's been a lot of increase in amyloid research, both from the biomedical point of view and from material science point of view. They have a lot of interesting properties, aside from the destructive properties that they exhibit, because they can actually, because of their fibrillar structure and the fact that they're quite ordered, they can generate a lot of materials with interesting properties, both mechanical and biophysical. Here in the image you see what these fibers actually look like in a diseased cell. Another reason that amyloids have been researched so heavily in the last few years is that there's been an increase in techniques, improvement in techniques to discover their atomic properties and other properties. Now, in addition to amyloids that cause diseases, there are some amyloids that have recently been found to be part of the normal cell cycle. And we call these amyloids functional amyloids. So they're being potentially non-toxic as opposed to disease-related amyloids. They can be the target of biomaterial design that potentially can be used in biomedical research, in green industry, and in many other applications. And this has been the focus of our research here. So we selected 2 short peptides that have been shown in a recent paper from 2009 to generate functional amyloids. One of them is called arginine vasopressin, and the other one is called neuromedin K. You can see their sequences here. They contain about 9 or 10 amino acids. We selected them, among other reasons, because they're very short, and therefore we can perform a full conformational search on them in a reasonable amount of time, unlike bigger protein molecules where this problem becomes computationally prohibitive using fully atomic MD simulation. So what we want to do is to perform a conformational search on these peptides, first of all in isolation, and then when they're conjugated to polymer molecules, and to investigate their conformational properties to see if they actually do have a tendency to self-aggregate, and then see what other applications, what other things we can do with them. What we did here is to actually combine simulated annealing with molecular dynamic simulation. Simulated annealing is a technique where we actually heat the system very, very quickly and then cool it down gradually in order to escape local minima in the potential energy surface. And that's how we can do a more extensive conformational search that doesn't necessarily depend on the initial conditions. So this is actually another application of molecular dynamic simulations where we can do a relatively fast conformational exploration of peptides and see what preferences, what structural preferences they have. And so if we run this process a number of times from different starting points, we can collect multiple low energy conformations that provide, hopefully, a reasonable sampling of the conformational space of those peptides. I should just note that this is not very feasible for larger proteins. And because of the small size, the 9 or 10 amino acids that these peptides contain, we can actually run using a supercomputer of 64 processors, multiple simulated annealing processes from different point of views, and we can provide a relatively good overview of the conformational space. So just a few details about the actual MD protocol. So the peptides were represented explicitly at the atomic level. The solvent was also explicitly represented. So we had approximately 3,000 to 4,000 solvent molecules per system to generate a solvent box of reasonable size. So overall, we had approximately 10,000, a little bit more than 10,000 atoms per system. So we used quite a standard protocol for the MD simulations. So first of all, we did energy minimization of the system using steepest descent to resolve initial clashes. We heated the system to 500 K using NVT, which is constant volume simulation. Then we did an initial equilibration for 500 picoseconds at room temperature, again in constant volume, followed by another 500 picoseconds of equilibration in constant pressure, so that the solvent box could adjust its size and change its volume to reach optimal density. Because the way we modeled the solvent box in the beginning is not necessarily the ideal optimal packing of the water molecule. So, and then we were ready to start the production runs. So first of all, we took the system and heated it to 900 K fast. And then gradually, very slowly cooled it down to room temperature. And each one of these cycles was run for 10 nanoseconds. And then we collected the conformations in each one of these cycles, low energy conformations. And after that, we took those low energy conformations that we collected and used them as starting points for new cycles of simulated annealing MDs, so that we can actually not being restricted to our initial starting point, which is actually pretty random. Because in this kind of peptides, we don't really know what the structure, what the stable structure might be, because there's not a lot of experimental data about it, and such small peptides usually tend to have more than one stable conformation. Okay, so just to be able to cover as many local minima as possible. And the last two stages were run in constant volume as well, using NVT. So overall, in this particular work, we ran 5 cycles and collected approximately 25,000 conformations per peptide. So the way we generated the conformations was using the AMBER molecular dynamic package, which uses a force field called FFO3. And the simulations were conducted using NAMD and AMD, which is a highly scalable molecular dynamics simulation program, which was developed by Klaus Schulten's group in the University of Illinois at Urbana-Champaign. And the nice thing about this program is that it can accept various force fields. Like AMBER, it was actually built originally with charm in mind, but it also accepts other force fields. And it's highly scalable, so you can run it on a supercomputer and it will scale very nicely up to quite a large number of processors. In this particular case, again, we ran with 64 processors, but it could quite easily scale up more on bigger systems. The simulation boxes were orthorhombic, which means it's actually a solvent box with these dimensions. So we actually surrounded each peptide by enough solvent for it to model infinite dilution with periodic boundary conditions and ionic strength to emulate physiological conditions. And we used the TIP3P solvent model, which is part of the AMBER force field to model water molecules. So what you see here in these two images, at the top, you see one of the peptides embedded in the water. And in the bottom, you see a zoom in on the molecule, and you see the water molecules surrounding it on the side. So it's actually, you can imagine a big water box with a molecule swimming in it. And the MD simulation is what would happen if you actually looked at this molecule over a period of time with its changing temperatures. So what I want to show you now is the trajectory of one of these conformational searches, just to show you what it looks like. And the display is done by software called VMD, which is also developed by the same group that developed NAMD. It actually stands for Visual Molecular Dynamics, so it allows you to visualize and analyze molecular dynamics trajectories. So what you see here is just a molecule. I removed the solvent box and the hydrogen atoms just for clarity. And this movie that you see here shows you the evolution of the structure of the molecule over a period of time. So you see this molecule actually swimming in the water that you don't see, but you see the molecule. So you see actually the trajectory which reflects the conformational preferences of this particular molecule over a period of time when we run the simulation. Okay, so you see the change of the molecule structure over time. So for those of you who are not familiar with visual representation of proteins, the lines here are covalent bonds between atoms. Carbon atoms are depicted in green or cyan. Nitrogens are in blue, oxygens are in red, and sulfur atoms are in yellow. And again, I omitted the hydrogen and the water molecules.